Hello, this will be a brief overview of biomarkers for the FDA Regulations class of the Regulatory Science Certificate Program in the College of Public Health of UAMS. I will discuss definitions of the word biomarker, continue with basic approaches for validation and interpretation of biomarkers, and end with a brief overview of their application by the FDA. There are many definitions of the word biomarker, but in 2015, a joint meeting of representatives of the National Institutes of Health, or NIH, and the FDA came up with this, which is available in their Biomarkers, Endpoints, and Other Tools, or BEST glossary. A biomarker is a defined characteristic that is measured as an indicator of normal biologic processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. By that standard, almost any endpoint of a study could be viewed as a biomarker. For example, in a preclinical rodent study, the time it takes for a mouse to find its way to a platform in the middle of a vessel of water and a swim test could be considered a biomarker of neurobehavioral effects of a novel drug or other xenobiotic. Or, a patient rating her pain level on a visual scale could even be considered a biomarker. But the group realized that was perhaps a bit too broad and added a qualifier. A biomarker is not an assessment of how one feels, functions, or survives. And that pretty much leaves us with lab tests, like blood and urine tests, and imaging, like X-rays, CT scans, and so on. So this is what I think it really boils down to. I think this is both more specific and more succinct. A biomarker is something in blood, urine, body fluids, or tissue that is measured in a laboratory or similar setting as an indicator of biology. Now, what qualifies as a tissue can be a bit squishy. We don't normally think of cell culture endpoints, for example, as biomarkers, but they're certainly important to consider in preclinical testing, and many of the principles that we will discuss here can readily be applied to those endpoints. For an example, please read the letter from the journal Toxicological Sciences that I wrote that is part of the class notes before this presentation. Now, the NIH FDA Joint Group divided biomarkers into 5 basic types, which I will briefly describe and provide classic clinical or regulatory examples for. First, there are biomarkers of diagnosis. For example, glomerular filtration rate, or GFR, an indicator of kidney function, can be estimated from measuring creatinine in serum. And any value less than 90 or so in a healthy young person, or 60 in an elderly person, would be considered a sign of potentially serious kidney disease. Next, there are biomarkers of monitoring. And there are really 2 types here. One is monitoring of disease. For example, measuring thyroglobulin levels as a marker of surgical success after removal of the thyroid gland in patients with thyroid cancer, or as a marker of recurrence after more general treatment of thyroid cancer. Patients without thyroid tissue should stop producing thyroglobulin. And 2 is monitoring for exposure. For example, measuring metabolites of environmental carcinogens in urine of a volunteer subject. Next, there are biomarkers of response. One example would be measuring the International Normalized Ratio, or INR, a biomarker of blood coagulation capacity, in patients being treated with the anticoagulant warfarin to ensure the warfarin is working. Next, there are predictive biomarkers. Predictive biomarkers are those that you measure in an individual before onset of disease or a specific treatment to determine the likelihood of disease or of either danger or success with that treatment. For example, before treating an HIV-positive person with the antiviral drug abacavir, they are often tested for a particular immune system genotype called HLA-B5701 to determine the risk of an adverse allergic drug reaction, such as a skin rash or liver injury. And finally, there are biomarkers of prognosis. These are markers that you would measure in an individual after onset of disease or treatment to predict the likely outcome of that disease or treatment. For example, C-reactive protein, or CRP, can be measured in patients with cardiovascular disease to predict the risk of complications or even mortality. Before moving on, you'll notice that I use the word predict when talking about both predictive and prognostic biomarkers. There's always confusion over those terms, and many people use them interchangeably. But in general, you should remember that prediction is done before something happens, and prognosis after. Now, before a biomarker can be used, it must be evaluated. This process is typically referred to as validation. Generally, a validation consists of 5 analytical components designed to ensure the accuracy and repeatability of the testing method, and 3 clinical components designed to enable interpretation of the test results. The analytical components can be remembered by the acronym PARS, which stands for Precision, Accuracy, Range, Sensitivity, and Specificity. Precision is the repeatability of test results. A test that is precise will produce roughly the same result or value for an analyte from a given animal or patient specimen every time it is measured in that specimen. That's illustrated here on the right by the precise target. Imprecision is due to error introduced during sample handling, sample preparation, test performance, and random error. Accuracy, on the other hand, is closeness to the actual value. Due to things like imprecision, all biomarker values are really just an estimate of the actual value in the sample, which is impossible to know with total certainty. Range, in this case, refers to the linear range of the test. Due to limitations of the instrument used to measure the biomarker, combined with other factors such as recovery of the analyte during sample preparation, there is usually a finite range of values for the biomarker over which the test is actually accurate, shown in the blue space on the curve on this slide. This is known as the linear range of the test. Next, sensitivity is essentially the lowest value for the biomarker that can be measured both precisely and accurately. And finally, specificity is the likelihood of interference in the accurate measurement of the biomarker. This is a bit harder to define, but one example would be antibody-based tests for amphetamines that also tend to detect the common gastroesophageal reflux disease treatment, ranitidine, which can lead to falsely elevated or false positive test results. Clinical components of the validation are reference range, which is typically the 95% confidence interval of values obtained from a population of healthy animals or patients, though it should be noted that many biomarkers are interpreted on the basis of a cutoff called a medical decision limit rather than a reference range. Clinical sensitivity, which is the percentage of a population with a condition or outcome of interest, such as a disease or an exposure, that have a positive test result, meaning either a result outside the reference range or above the medical decision limit, and clinical specificity, which is the percentage of a population without the condition or outcome of interest who do not have a positive test result. And the clinical components of the test validation are especially important because they are the foundation of interpretation. Before we get into this, I want to add that this is not a math class and you will not be tested on your ability to perform these calculations. But this is critical knowledge for anyone who might be in a position to interpret test results, whether during drug testing for regulatory purposes, or even in your own personal medical care. So you should understand these concepts. Now, we can calculate sensitivity and specificity very easily using a 2x2 table as shown here. If we have a population of 100 patients, 40 with the disease and 60 without, and the test results shown in this table with positive biomarker results on the left, negative biomarker results on the right, patients who are positive for a disease, in this case some cancer in the top row, and patients who are negative for that cancer in the bottom row, we can plug these numbers into the equations for sensitivity and specificity shown at the bottom. So this is simply true positives, that is, patients who have a positive biomarker result and have the disease of interest, divided by true positives plus false negatives, that is, patients who are positive for the disease but have a negative biomarker value. And that gives us sensitivity. In this case, it's 0.75 or 75%. Similarly, we can calculate specificity by plugging in the value for true negatives, that is the number of patients who do not have the disease and had a negative biomarker test result, divided by the sum of true negatives plus false positives, that is patients who did not have the disease but had a positive test result. And when we do that, we get a value of specificity of 0.92, or 92%. The problem with sensitivity and specificity is that they do not account for prevalence of the condition of interest. This test will always detect 75% of the patients with the disease, whether 40% of the population has it or only 4%. However, if you think about it, if the disease exists in only 4% of the population, then the starting probability of any given person in that population having the disease drops precipitously, and the likelihood that a positive test result is actually a false positive increases. In other words, the more rare the condition for which you are testing, the greater the probability of false positive test results. To deal with that, we can also calculate predictive values as demonstrated here. And to do that, we simply change one number in each equation. For example, instead of calculating sensitivity using true positives divided by the sum of true positives and false negatives, we can calculate the positive predictive value by dividing true positives by the sum of true positives plus false positives. This gives us a value of 0.86 or 86%. The negative predictive value is calculated from true negatives divided by the sum of true negatives plus false negatives. And in this example, we get a value of 0.85 or 85%. And these values are far more useful than the more commonly reported values for sensitivity and specificity, because predictive value is the percentage of patients with a positive or negative test result who actually have or don't have the disease. And that's really what you want to know when, for example, you're trying to diagnose a patient. That is, if a patient has a positive biomarker test result, what is the probability that that patient has the disease based on that positive result? That's the information you can obtain from a positive predictive value, or PPV. Similarly, the negative predictive value gives you the percentage of patients without a positive result who do not have the disease. And we can show why this is important simply by changing the prevalence of the disease in this example. You will recall that we calculated that sensitivity and specificity were 75 and 92% respectively. We also said that 40 patients out of our population of 100 have the disease, which gives us a prevalence of 40%. And from that we arrived at PPV and NPV both around 85%. Now let's keep our sensitivity and specificity the same, but change our prevalence to 4%, which is tenfold lower. And now we're working with proportions rather than absolute values, but the principles are the same. By multiplying the prevalence, 0.04 or 4%, by the sensitivity, which again is the proportion of patients with the disease who will have a positive biomarker result, we see that our true positive rate is 3%, or 0.03. That's in the upper left box in the 2x2 table. To determine our false negative result, we then simply subtract 0.03 from our prevalence of 0.04 to get 0.01. That's on the right side of the box. To determine our true negative rate, we then multiply the proportion of patients without the disease, 0.96, by the specificity, 0.92, and we get 0.88 in the lower right part of the box. And finally, we subtract the prevalence of disease, 0.04, and the true negative rate from 1, or 100%, to get the false positive rate of 0.08 in the lower left portion of the box. We can then plug these numbers into our equations for PPV and NPV, and we can see that our PPV has dramatically dropped to 0.27 or 27%, while our NPV has increased to 0.99 or 99%. This makes the test somewhat more useful to rule out the disease for a given patient. That is, if the patient has a negative biomarker result, we can be 99% sure she does not have the disease. but it's far less useful to make an actual diagnosis. The probability of disease based on this result alone is only 27%. Again, you will not be asked to do this math for this class. However, the concepts illustrated here are critical. Simply put, when evaluating the utility of a biomarker, prevalence is supremely important. And this applies to any type of biomarker, with some exceptions. Obviously, it applies to biomarkers of diagnosis. It also applies to biomarkers of prediction and prognosis, where you're testing for a disease outcome rather than for the disease itself. And you can apply this to monitoring for exposure, where instead of testing for disease, you're testing to determine if a person has been exposed to a carcinogen, for example, or some other environmental toxicant. If you need to, please go back and replay the last 6 slides covering clinical sensitivity, specificity, and predictive values to be sure you understand this material. It is very important. Now, predictive values are not without challenges. Determining PVs requires prospective studies in which a real-world population can be followed in order to determine true prevalence and real-world use of your biomarker of interest. Fortunately, reasonable alternatives to predictive values are available. I will not discuss those in detail here, but I've listed the 2 most common alternatives, which are likelihood ratios and post-test probabilities. So why does the FDA care about biomarkers? Well, there are many reasons. As you might expect, they can use biomarkers to identify potential for drug toxicity during preclinical and clinical trials. Biomarkers can also be used by clinicians to identify patients for clinical trials and ensure proper diagnoses. Next, biomarkers of response can be used to determine response to treatment in patients during clinical trials. The FDA is also very interested in the development and validation of new biomarkers that can provide more information than the current standard biomarkers. For example, a phenomenon frequently encountered during clinical trials is transient elevation of the standard liver injury biomarker alanine aminotransferase, or ALT. In many cases, the FDA is reluctant to approve drugs that show any evidence of potential for liver toxicity. However, it is now known that many drugs can cause minor, transient elevations of serum ALT values that do not portend clinically significant liver injury. So the FDA and pharmaceutical companies have a keen interest in development of biomarkers to predict real liver toxicity. Other areas in which the FDA is interested in development of novel biomarkers include markers of acute kidney injury, markers of cardiotoxicity, and markers of neurotoxicity. Although any data for any biomarker that may be useful for interpretation of results from preclinical and clinical trials can be included in a submission to the FDA for a new drug or medical device, there is no guarantee that the FDA reviewers reviewing the submission will acknowledge or accept those data. One way to improve the likelihood of acceptance is to use qualified biomarkers. Through the FDA Biomarker Qualification Program, or BQP, interested parties, called requesters, can submit data that supports the utility of a particular biomarker for a particular use. The FDA will review those data and decide whether or not they agree. If they agree, the biomarker is said to be qualified. This process progresses through 3 steps, beginning with a letter of intent submitted by the requester that describes the biomarker they intend to use, the context in which they intend to use it, or context of use, COU, and how the biomarker will be measured. If the letter is accepted, the requester will then submit a plan for qualification which describes the studies they will do to test the utility of the biomarker for their COU. Finally, the requester will perform the studies and submit the results to the FDA as part of the final qualification package. More information about the BQP is available through the links and the course materials for this week. And that concludes your very brief lesson in biomarkers and their regulatory use. Thanks for your attention.